Great. Thank you very much, Marek and Marianne and Dov and the other organisers and the Menarini Foundation. It's an absolute pleasure to be here at this wonderful meeting. So I'm going to talk to you about a number of our preclinical models, in particular a mouse model of Huntington's disease, a couple of mouse models of schizophrenia, and then I'll talk about our work on paternally mediated intergenerational epigenetic inheritance. We have got some further evidence going to F2 transgenerational epigenetic inheritance through the paternal line, but I won't discuss that. So to introduce Huntington's disease, I first want to talk about it as a monogenic tandem repeat disorder. So it's one of the more common of over 50 now monogenic tandem repeat disorders. But to understand this, we need to understand that approximately half of the human genome consists of repetitive DNA, or repeatome. And within that repeatome, there's approximately 1.5 million distinct tandem repeat sequences, about which we know almost nothing. The one that causes Huntington's is a CAG repeat, effectively a genetic stutter that encodes a tract of glutamines, a polyglutamine tract, and if you have more than 35 and usually 40 or more, you'll develop Huntington's disease. And this makes it extraordinary with these kind of monogenic disorder. Hopefully no one from this room is from a Huntington's family. But if you are, the only thing that separates you from someone who will die horribly from Huntington's, usually over a couple of decades, is that in the 3 billion base pairs of DNA in their genome, the only difference is a handful of extra repeats that makes them die horribly of Huntington's, and you hopefully survive a lot longer. So one thing that interests this audience is all of these potential functions, which we know very little about, of these other 1.5 million tandem repeats. Many of them are located outside of coding regions, and we think many of them have evolved related to epigenetic regulation because they can undergo different DNA modifications and chromatin changes, etc. One of the exciting things in this field, just in the last couple of years, is using whole genome sequencing with autism, including trios, so as to pick up de novo mutations. It's very clear that there are many different tandem repeats, when mutated, can give rise to autism with different levels of penetrance, obviously still with environmental factors and epigenetics at play, but this has really exposed this field. And I'd suggest that you keep an eye on schizophrenia and other complex disorders. There's tandem repeat association studies in the pipeline, and I would view this potentially as the tip of the iceberg. Keeping in mind, we've heard a lot about GWAS. Genome-wide association studies, just over 15 years, have been SNP-based, and therefore the single nucleotide polymorphisms mutate far more slowly than the average tandem repeat, and therefore SNPs are often not going to tag for tandem repeats, which are far more mutable. And so many of these tandem repeat mutations and polymorphisms will not be identified by SNP-based GWAS. They need whole exome, ideally whole genome studies with appropriate bioinformatics. So focusing now on these now greater than 50 tandem repeat disorders, and you'll be familiar with ones like Friedreich ataxia and Fragile X syndrome, which do seem to have epigenetic changes as their primary driver. But Huntington's is the most common of at least 9 polyglutamine diseases, and the model I'll be showing generates toxic gain of function as its primary mechanism, although we are pursuing the possibility of change of function, that is, the polyglutamine tract evolved a normal function. When you expand it, it distorts the normal function, and therefore we need to understand the normal function to understand Huntington's, and our data and that of others suggests that Huntington's could have developmental components. So it might be developmental changes overlaid on neurodegeneration that ultimately give rise to Huntington's. Again, with these multiple monogenic tandem repeat disorders and the tandem repeats that are mutated within them, that's given a lot of insight into the way that these tandem repeats are associated with epigenetic changes. For example, it appears that the expansion of the FMR1 tandem repeat causes epigenetic changes to turn off FMR1 and hence cause Fragile X syndrome, and there's different repeat lengths that can cause different Fragile X syndrome-like disorders, or in fact a neurodegenerative version of it, FX-TAS, with a different repeat length. The epigenetic changes in Huntington's we think are downstream of the polyglutamine tract, and we've been working more recently on non-coding RNAs. I'll talk a little bit about that later on, but not in the context of Huntington's pathogenesis specifically. So here's our model. We have the advantage with this kind of autosomal dominant disorder that we have a model with exquisite construct validity, with very clear face validity, which we've now demonstrated over 2 decades. And in some of the features, like we showed we could model depression in an animal model of HD. You can also show predictive validity. You can give them antidepressant, for example. So it's a great advantage in knowing you have a preclinical model that's very accurate, so that when we do things in this model, for example, test drugs and other interventions, we're more confident when we go to the clinic that this will translate. And just like a Huntington's family, where one of the parents has the Huntington's mutation and the other doesn't, generally, we breed from male HD mice and breed them with control female mice, and the only difference in the offspring is that half of them, just like a Huntington's family, have the Huntington's mutation. The other half are perfectly strain-matched, randomized litamate controls, but they lack the mutation, but are otherwise perfectly strain-matched. And we can show the face validity, not just with behavior, but also with these changes in the brain, shown here, coronal section of the HD and wild-type mice with degeneration of the striatum and cortex that very closely reflects clinical Huntington's. So here's this environmental enrichment that was beautifully introduced by Marek earlier on. And so essentially, for those who don't work with animals, what we do is, again, in a randomized fashion, in these original experiments, we took them at weaning. In this case, it was 4 weeks postnatal. And we randomized HD and wild-type mice into standard housing conditions, which is group housed, soft bedding, unlimited food and water, but very little apart from nesting material in the cage, versus environmental enrichment, which adds these enrichment objects to increase novelty and complexity, to increase sensory stimulation, cognitive stimulation, and opportunities for physical activity. And this data was from my first PhD student, Anton van Dellen, and what we were able to show, and it really took us by surprise, was that those HD mice that had the environmental enrichment from weaning showed a dramatic delay in onset of motor symptoms. So looking at the standard housed here, by about 5 months of age, 100% is showing this motor deficit. This is important because in Huntington's, clinically, 95% have adult onset. There's 5% juvenile HD, and therefore this is part of having a very, very accurate model. However, if we just gave them environmental enrichment, we had a dramatic delay in motor onset. And this took us by surprise at the time. At the time, there were textbooks and people would talk about Huntington's as 100% genetic, the epitome of genetic determinism, nothing you could do. So this was evidence that even something that supposedly is 100% genetic, you can shift it with environmental factors. An important aspect here too, because I have been to meetings whereby people have animal models where a drug or something's working and they go to the clinic and the clinical trial fails, and people say, well, let's just give it up on the animal models and we'll all work on iPS or organoids. And that's the case. But I think part of it is, if you have an accurate model, and we have the advantage in Huntington's of this genetics that allows us to have an accurate model, then it can translate. And indeed, this is robust as well. Colleagues with independent models of Huntington's have independently replicated this many times. We've been able to show not just delay in motor onset, but delay in cognitive deficits modeling dementia, and delay in the depression-like signs that we'd been able to discover previously in this HD animal model. And so I first presented this in Boston in front of Nancy Wexler. She's been a leader in the Huntington's field, an extraordinary woman. Her mother died of Huntington's. Her father drove enormous philanthropy for Huntington's. Her sister Alice has written a couple of famous books on Huntington's. And in fact, we knew because of her mother that she was 50% at risk, and Nancy's a dear friend, and we'd go to conferences and people would, you know, obviously in Huntington's a lot of people won't talk about their genetic status, but we've gradually seen that Nancy, you can see Korea, it's very clear, and we've seen Nancy become symptomatic, and so it's a type of field where you end up with a lot of friends and knowing HD families and knowing that you know, half of them are going to die of this disease if we can't develop a treatment, which is very motivating. So my colleagues in Melbourne, led by Martin Delaticchi, took our mouse work and, well, firstly I should say that Nancy followed up our work in the Venezuelan kindreds and was able to show for that variance in motor onset for a given CAG repeat length, it's obviously genetic and or environmental modifiers. And the kindred study that they did showed that it was more environmental than genetic, and then the retrospective epidemiology done by Martin and colleagues based on, I was sending them all the papers on Alzheimer's and dementia and so on, suggested that people who were more passive and less active for a given CAG repeat length had earlier onset. And now there are prospective intervention trials and so on occurring in the field. So getting back to the mice, having discovered this at a behavioural level, we then wanted to explore mechanisms, and therefore at the end of these behavioural experiments, we'd take these tissues, the 4 groups, HD and wild type mice randomised into environmental enrichment standard housing, and over many years we've gradually worked our way through not systematic epigenomics in terms of methylation, but more recently starting to look at non-coding RNAs, but also mRNA transcriptomics, proteomics, phosphoproteomics I won't talk about today, and some metabolomic work as well. So focusing on the synapse in terms of, we think that Huntington's is at least partly a synaptopathy, a disorder of synapses, and we've been able to show particular proteins, for example, just focusing here on the PSD95 protein, which is very important for these forms of synaptic plasticity. And at an age when we can show onset of the cognitive deficits modelling dementia, there's a big deficit in this PSD95, but environmental enrichment rescued it. So this is the type of molecule you might want as a therapeutic target. A disease has modified the molecule, environmental enrichment has moved it back to normal while correcting cognitive, affective and motor symptoms, and therefore we can use this paradigm to identify therapeutic targets. Now it raises a question of why at the protein level these molecular changes might occur, and there's a whole range of potential reasons, but one of them we think is epigenetic, that one of the ways, and Marek mentioned this, that one of the ways that enrichment can work, we think, is epigenetically because it has very long-term impacts. So as soon as you see very long-term impacts of environmental intervention, one possible mediator is epigenetics, because it is one way that you can induce long-term changes. And this study suggests, at least in the dentate gyrus of the hippocampus, that environmental enrichment in wild-type mice can delay aging, keeping in mind that Huntington's, like Alzheimer's, Parkinson's, motor neuron disease, the biggest factor is aging in any neurodegenerative disease, and then you've got genetics and environment overlaid on aging as other risk factors. So we think epigenetics is involved in the therapeutic effects of environmental enrichment. Going back to the dentate gyrus here, it's a very interesting region because it's a region in the adult brain where neurogenesis occurs throughout life. And so we've been interested in this for a number of reasons. So we were able to show originally with Roger Barker in Cambridge that these Huntington's mice have deficits in adult hippocampal neurogenesis. And the interesting thing here is on the left here in rodent models, there's a whole heap of models whereby you see decreased adult hippocampal neurogenesis coupled with depression-like or cognitive deficits in the rodent model. We were able to add Huntington's to this list. We've been able to show serotonergic changes, BDNF signaling changes, glucocorticoid signaling changes, which are associated with stress responses on that side of the coin, if you like, with Huntington's being the example here. On the other side, on the right side, are these factors associated with increased adult hippocampal neurogenesis in rodent models, often coupled with antidepressant-like effects in the rodents and cognitive enhancement, hippocampally mediated at least in these. And on this side, we have environmental enrichment, we have exercise or physical activity alone, and these selective serotonin reuptake inhibitor antidepressants, or SSRIs, among the most commonly prescribed fluoxetine, sold as Prozac, or sertraline, sold as Zoloft. All right, so for all of these reasons, this took us into an interest in seeing if we could model depression, keeping in mind that of psychiatric symptoms in Huntington's, depression is the most common. Approximately 50% of those with the gene mutation, even if they don't know they have the mutation. And the mice don't know they have the mutation, so it gets away from any possible psychosomatic effects of knowing you have a gene mutation that causes a fatal disease. And so we're able to show on 4 independent tests of depression-like behaviour that these female HD mice show depressive-like behaviours. Now, on first principles, my lab has always looked at both male and female rodents. It just seemed instinctively the thing to do because all of the disorders we study occur in both sexes. But when I looked around in neuroscience, a lot of people were only studying male animals, and the argument was the estrous cycles are confounded and so on. But it's very easy to check that confound because whenever you power your experiments equally in males and females and look for variance, if the estrous cycle is adding variance, you should see it pop out of the error bars. And we've almost never seen, the things we measure, we've never seen more variance in the females. Admittedly, we've not been focused on sex hormones, etc. And therefore, by studying both sexes, we discovered this. We would have missed this completely if we'd just been studying male HD mice and their wild-type littermates. So the important thing here is that we have face validity for depression, that the sexual dimorphism fits with clinical depression in the general population. It's about 2 to 1 females to males. So we're modelling the degree of sexual dimorphism in the general population, which we think kind of match it in the HD population. We think it's somewhat similar. That face validity, but finally we have predictive validity. So we give not just acutely, but chronically, if we give 2 weeks of sertraline, an SSRI marketed as Zoloft, then we can have antidepressant-like effects. And this is on the test of anhedonia, saccharin water versus just water, and mice will drink 90% of their fluid from the sweet water. The female HD mice on 3 other tests are showing depressive-like behaviours. They also show anhedonia. They drink less sweet water, and the antidepressant drug brings them back to normal. We can also show that environmental enrichment has antidepressant-like effects in these female HD mice. And moving on, we then we just gave running wheels in the cage. We wanted to see what exercise alone could do as part of the combined environmental enrichment effect. And overall, what we see is that the combination of cognitive stimulation and physical activity with environmental enrichment has a bigger effect than just wheel running exercise alone, but exercise alone does have significant effects. And this is interesting as well because exercise clinically can have antidepressant-like effects in clinical populations. We also find that the female HD mice, I won't go into details, but at the time they're showing depressive-like behaviours, they show dysregulation of the HPA axis, and there are physiological changes in response to acute stress in these female HD mice that can be ameliorated by this wheel running exercise. We're wanting to know how the environmental enrichment exercise works, and we've been doing a lot of molecular work. We're pursuing this at the moment, that there are a whole bunch of small non-coding RNAs that Kui Chen spoke about the other day. MicroRNAs are an important subset, and we find many of them are changing in the hippocampus due to exercise, and we're currently pursuing them. So bringing this together, thinking about these environmental modulators and molecular mediators, we think a subset of these molecules that are changing with these therapeutic environmental interventions might be targets for environmemetics. They will be novel therapeutics which mimic or enhance the beneficial effects of cognitive stimulation and physical activity. So moving now just briefly to a more complex disorder, schizophrenia. The genetics is extremely complex, but we've made a lot of progress in terms of the genetics. So thinking about the genome, but also the epigenome, of course, is highly relevant. And then the environmental factors, or enviromes. Other people use exposome, it's another term that we need to think about at different stages of life and how these might combine to lead to schizophrenia symptoms, but also the changes in brain maturation and neural circuitry that give rise to this complex disorder. We've been able to show that PLC beta 1 in mice, when it's knocked out, the protein's not present during development, that these mice have disrupted experience-dependent cortical maturation, and we think maturation in other parts of the brain as well. And this took us into interest in this pathway, and we think particularly metabotropic glutamate receptor 5 signaling onto PLC beta 1 is of relevance to the pathogenesis of schizophrenia, as are many other pathways, but also an important pathway in experience-dependent maturation of the brain. I won't dwell on this data, which is really just showing that using synaptoneurosomes, that stimulating mGluRs, and we think it's mGluR5 in particular, when you've knocked out this gene PLC beta 1 from the cortex, you lose signaling through that pathway. We phenotyped the mice on a whole bunch of behavioural tests, and on the right you can see these schizophrenia-like behavioural endophenotypes, that is face validity. On the left are the clinical endophenotypes of schizophrenia, and these are nicely aligned, so we have good face validity. And the bottom, with the antipsychotic drug clozapine, we've been able to show predictive validity. And very interesting, Merrick mentioned potential epigenetic effects of clozapine. Obviously, like antipsychotics, they're dirty drugs, they have multiple mechanisms of action, but we are interested in the idea that clozapine could have some of its actions, perhaps, by at least one epigenetic pathway. That was all under standard housing, and so we did the environmental enrichment experiment. And the important thing here is KO refers to knockout. It's actually with PLC beta 1, you're kind of knocking out about 5 important pathways that we think are relevant to schizophrenia. So if you want to model a psychiatric disorder, you actually have to have this type of mutation, ideally, that's disrupting multiple pathways involved in brain development. And SH, standard housing, is really a sensory motor deprivation state. Environmental enrichment is closer to the average human level of cognitive stimulation and physical activity, and only when you combine a negative genetic factor with a negative environmental factor do you get a fully penetrant schizophrenia-like phenotype. And this is exactly how you want to model a psychiatric disorder. We've also been able to show this upstream. When we knock out mGlu5, we also get a schizophrenia-like model, but it's a bit more subtle, so less severe when we've just knocked out the one receptor as opposed to PLC beta 1. But we also have predictive validity. When we give them chronic clozapine, keep in mind also that patients often take a couple of weeks to respond to these drugs. So chronic clozapine has positive impacts on these mice as well. And we can show changes in brain development that match these schizophrenia-like endophenotypes. In this case, we've been able to show that environmental enrichment corrects this cognitive deficits. This is on the Morris-Water maze. This is important because antipsychotic drugs, A, they're dirty, they have terrible side effect profiles, and they also don't correct the cognitive deficits, which are among the most devastating symptoms of schizophrenia. So if we can work out how environmental enrichment corrects these cognitive deficits in a schizophrenia model, perhaps via environmentic type mechanisms, then we'd have potential breakthrough in schizophrenia, which is one of the most devastating of all human disorders and is largely very poorly treated. Now we're looking to translate all of our work, and we have a number of translational tools. Have a look at these touchscreens. On the left you see an iPad or touchscreen up the front. The mouse touches its nose and then comes back on the left to get a strawberry milk reward. So it's effectively CANTAB neuropsychology for mice. If you look on the right, the 2 stimuli will come up on the iPad or touchscreen. The mouse will touch its nose on the screen to make a choice. Touches left. This time the light comes on. The mouse knows it's wrong. It doesn't look for the strawberry milk reward. And this is directly analogous to CANTAB or human neuropsychology batteries. So the idea being that if we have an intervention like a drug trial that works on these tests and we take it to the clinic, it's more likely to translate successfully. We've been able to show that environmental enrichment can, on these touchscreen tasks, can enhance cognition in wild-type mice. We've been able to show that mGlu5 mice have deficits on these touchscreen tasks that closely model the kind of cognitive deficits you see in clinical schizophrenia. So getting back to Waddington and the epigenetic landscape, we're thinking about these gene-environment interactions as the brain being experience expectant. So back to Waddington, the idea that development, and we're interested in brain development, being normally canalised via evolution and genetics. However, decanalisation can occur due to absence of expected instruction. So think about this DOHAD concept that Marek has introduced, the idea that globally we've moved a long way from what we've evolved to see in utero, throughout postnatal life, diet, poor diet, Western diet, sedentary behaviour, high levels of chronic stress, toxins, you name it. And therefore, these absence of the expected instructions can decanalise development, which we think may be relevant to disorders like autism and schizophrenia, but also due to an unexpected instruction like a major stressor in utero, it may be relevant to both autism and schizophrenia. And then in adolescent critical periods, it may be particularly relevant to schizophrenia, which has later onset. Usually in males, late teens, early 20s, a slightly different profile in females. Moving on, and this Waddington landscape we think is totally relevant to this paternally mediated transgenerational epigenetic inheritance. Most of what I'll talk about is intergenerational epigenetic inheritance, but we have in some of these models, and others have shown, including Isabel Mansou in Switzerland, that the early life stress in male mice can carry through to F3 and F4. At least early life stress may have very long-term transgenerational effects. I won't dwell on these prior studies for the interest of time, but focus on our work, which took this stress model involving corticosterone. So it's very clean. The mice will drink corticosterone, the direct equivalent of cortisol in humans, a stress hormone. And so you can lift up their stress hormone levels in a very precise molecular way. And so we had the control males who had 4 weeks of drinking quart to increase their stress hormone. And then the others everything was the same, but they just drank water. And then we mated them with control females and looked at the F1 offspring. And what we found is that in the offspring, and this is sexually dimorphic, which we see on most of these paradigms, that the male F1 offspring had increased anxiety on these 2 independent tests of anxiety-like behaviour. We obviously wanted to see what was happening in the F0 fathers to their sperm and their epigenetics, and following on from the excellent work of colleagues. I won't talk about it today, but we've isolated the DNA from the sperm of the fathers and used Oxford nanopore sequencing to get whole methylome data, and we do have evidence that this paternal quart, increased stress hormone, can specifically change individual genes, their methylation in the sperm. But in terms of testable evidence, we think these non-coding RNAs, we're a bit more confident that they may be causally related. And I'll speak about our evidence that they're changing. So we isolated these non-coding RNAs, the small non-coding RNAs from the sperm. Kui Shen gave a lovely talk yesterday on his work that's been pioneering in this field. And we found that the increased quart or stress hormone caused many microRNAs to change in the father's sperm. And we could couple some of these because each microRNA can bind to the 3' UTR of multiple targets, so they're kind of master regulators. And if we looked in the hippocampus of the F1 offspring brains, we could see that particular microRNAs and their targets, in this case BDNF and IGF2, were altered in the offspring brain. Now that's correlation, but it's something we're still following up. In terms of how this might occur, we think that these changes might occur via extracellular vesicles, in particular epididymosomes, that might deliver these non-coding RNAs into the male germ cells and ultimately the sperm. And this is a hypothesis we put forward, but data has accumulated in the last 5 years to support this idea. And we've used other interventions. So if we just give the exercise to the males or have standard housing, then paternal exercise prior to conception, when looking at their offspring, can have changes in behaviour, particular cognitive and affective phenotypes that change in the offspring. Again, we looked at the F0 fathers, isolated the small non-coding RNAs from their sperm, and found specific small non-coding RNAs that were changed in the sperm, not just microRNAs, but these tRNA fragments that Kui Chen talked about yesterday, or tsRNAs, as he referred to, that are caused by paternal exercise. Now down here, this relates to what Marek was talking about. We've also looked at the fathers with paternal environmental enrichment with this former student, Shlomo Yeshurun, who's now returned to Israel. And he was able to show that the F2 mice were affected by the F0 father's environmental enrichment. So we have evidence of transgenerational effects with that particular environmental intervention. So putting all this together, these different environmental exposures in males may change the sperm epigenome in a broad definition, including non-coding RNAs. And the hypothesis is, and obviously we need a lot more human data, but that this could cause changes in brain development and maturation that lead to different cognitive, affective, and other behavioural traits in the offspring. There's a lot of rodent data now, but we absolutely need more human data in this space. Thinking about how these environmental exposures or environment could change the male germ cells and the sperm. Lovely work from Isabel Mansour's lab again in Switzerland showing that small molecules or metabolites in the blood could signal to the male germ cells, perhaps to cause some of these changes. And ultimately, we want to think about the transfer of this information. It might lead to epigenopathy, epigenetic contributions to human disorders. Or on the other hand, certain environmental factors might be able to induce a form of epigenetic resilience, and we need to understand these. Finally, a very recent study we did, following up work of many others, including Kui Chen and Margaret Morris from Sydney, had one of the first studies, was to show that the novelty here was we gave the Western diet, high fat, high sugar, during adolescence in the male mice, and then we bred them later on, and we did very deep phenotyping of the F1 offspring. And I'm just going to focus on, you can look at this published paper, but we had very interesting behavioural changes, but we also had a change in the gut microbiome of these F1 offspring. Now, I haven't referred to our other data. We discovered about 4 years ago the first evidence in Huntington's disease that the gut microbiota are changed, i.e. there's gut dysbiosis. In the genetic model of schizophrenia, we've discovered gut dysbiosis there as well. But in this case, we were very surprised that this paternal diet in fathers could lead, even though it's a subtle change in the microbiome, it could lead to a significant change in the microbiome of the F1 offspring. And the data from other groups suggests that these microbiota changes via the microbiota gut-brain axis could relate to the kind of behavioural changes that we showed in the F1 offspring. So this is an article from a number of years ago, You Are What Your Dad Ate. Obviously, all of these changes across diet, across stress, physical activity, this could have major repercussions in terms of public health. We need to understand this. We need to, if there is epigenopathy that can be transmitted through generations, we need to work out how to prevent that transmission. Once it's transmitted, we need to diagnose this epigenopathy and have these precision or personalised medicine approaches to be able to target it. So a lot of this comes back to the idea that we should never, ever dismiss new ideas out of hand. Lamarck was ridiculed. The fact is that Lamarck was not completely wrong, and we need to maybe think about a synthesis between Darwinian and Lamarckian evolution. I used to say we're all dealt a genetic deck of cards at conception we can do nothing about, but now maybe with CRISPR gene editing we can do something about it, but we're also apparently dealt an epigenetic deck of cards at conception. The red arrows on the right are indicating movement towards abnormal brain development. Brain and cognitive reserve may build in neuroprotection. We can build in functional compensation with positive factors. Negative factors like stress may lead to particular brain disorders, and positive interventions like environmemetics may help us get to this side of healthy brain maturation and aging. As a subclass of environmemetics, we're thinking about epigenetics. Moshe Ziff gave a beautiful talk yesterday on this epigene therapy. This may be part of it, but we're also thinking of small molecules that could target particular epigenetic components, thinking specifically here about beneficial environmental interventions that lead to epigenetic changes. And finally, this positive effects of exercise. We're thinking, how do we go after exercise mimetics? How do we mimic or enhance the beneficial effects of exercise to harness this therapeutically? So finally, I'd like to acknowledge and thank the wonderful people in my lab that did all the work. Just in this case, Terence, Emma, Shan, Karina, Thibaut, Ariel, Shlomo, and Annabelle, and their dedication and resilience through this last couple of years of the pandemic. And I'm happy to answer any and all questions. Thank you.